Welcome to Roadmap to Residency Evidence-Based Applications. Hello everyone, I am Abhiraj. Welcome to our Club to 50 Matchmaker sessions. So in this video, we will be discussing in detail about the cell signaling pathways. So cell signaling, they regulate the virtually all aspects of cell behavior, the cell proliferation, the metabolism, the organelle and cell movement, apoptosis, and whatnot. The modes of cell signaling are direct cell signaling, signaling by secreted molecules, and signaling molecules. Direct cell signaling is a direct interaction of a cell with its neighbor. Signaling by secreted molecules, which are signals, are transmitted over distant cells. The signaling molecules from one cell bind to receptors in other cells, range in complexity from simple gases to proteins. So endocrine signaling, so signaling molecule or hormones target cells at distant body sites. Example, estrogen is endocrine signaling. Paracrine signaling act on the neighboring target cells. Example, neurotransmitters. And autocrine signaling is response to self-signaling molecules like T lymphocytes, so that the proliferation in response to cytokines produced by the same cell. So classes of signaling molecules are hydrophobic signaling molecules, neurotransmitter, peptide signaling molecules, or eicosanoids. So hydrophobic signaling molecules, so they passively diffuse across the cell membrane like steroid hormones, vitamin D3, thyroid hormone, retinoic acid, nitric oxide, and carbon monoxide. So you need to remember these names. They all use intracellular receptors and they function as activators or repressor of genes. So neurotransmitter carries signals between the neurons from neurons to other target cell. So hydrophilic, so they can't cross the plasma membrane of target cells and they bind to the cell surface receptors. That's why. So the receptors are the ligand-gated ion channels and the neurotransmitter binding opens these channels. So peptide signaling molecules are the peptide hormones, neuropeptides, or polypeptide growth factors. They all use the cell membrane anchor receptors. So eicosanoids are the lipid signaling molecules, which include prostaglandins, prostacyclin, thromboxanes, and leukotrienes. Arachidonic derived from phospholipids or phospholipase A2. Peptide signaling molecules, so the cyclooxygenases catalyze the conversion. They are short-lived, thus act in autocrine or paracrine pathways. The platelet aggregation, inflammation, smooth muscle contraction, they all are through the peptide signaling molecules. So there are some signaling molecules, so there should be some signaling receptors too. So there are two types of main signaling receptors. They are the intracellular receptors and membrane integral receptors. The intracellular receptors include nuclear receptor family and they bind the hydrophobic signaling ligands. The conformation chains translocate to nucleus and they act as the transcriptional activators or repressors to regulate the gene expression and signal does not amplify. In case of membrane integral receptors, they bind the non-hydrophobic signaling ligands. There's conformational change that activates a phosphorylase cascade through kinases, which are the enzymes, and they might rely on secondary signaling molecules. The exponential signal amplification is through the kinase cascade, and the targets are transcription, replication, translation, cytoskeleton remodeling, and metabolic modulation. The intracellular receptors, or for example, the nuclear receptors. So nuclear receptor is a superfamily. So they are the transcription factors and have domains for ligand binding, DNA binding, and modulation transcription. For example, glucocorticoid receptor, which is bound to the HSP90 chaperones in the absence of hormone. The glucocorticoid binding displaces the HSP90, binds the regulated DNA sequences together with HAT coactivator. HAT is histone acetyl transferase. Thyroid hormone, so the hormone binding may alter the activity of the receptor. So in the absence of hormone, the hormone receptor is associated with a compressor complex and there is no transcription of a target genes. But whenever there is hormone binding, it replaces the co-repressor complex and then the transcription is activated. So membrane receptors, that is the G protein coupled receptors, are the largest family of cell surface receptors. And there are 7 membrane spanning alpha helicals. And there are various signaling ligands like hormones and neurotransmitters. The signals transmitted via guanine nucleotide binding proteins are the G proteins, and they have various targets and effector proteins. So this one is important. The receptor protein or tyrosine kinases. So humans have 59 receptor protein tyrosine kinases, and the signaling molecules like epithelial growth factor, platelet-derived growth factor, and other growth factors, and insulin, most importantly, function through the protein kinases. They phosphorylate their substrate proteins on tyrosine molecules. So ligand-induced receptor dimerization takes place. The ligand binding activates the cytosolic kinase domain, and then there is the autophosphorylation of receptor. And whenever there is autophosphorylation of receptor, there is increased in the protein kinase activity, and it creates the binding sites for other proteins that transmit signals downstream of the activated receptors. So there are some non-receptor protein tyrosine kinases, uh, but these are present in the cytosolic domains and have no catalytic activity. The cytokine receptor superfamily like interleukin-2 and erythropoietin act on these, and the cytokine receptor function in association with non-receptor tyrosine kinases like Janus protein kinase or the JAK kinases. The cytokines regulate the development and differentiation of lymphocytes during immune response. Now, the intracellular signal transduction pathways. So we have talked about the molecules, we have talked about the receptors, and now let's talk about some pathways. So intracellular signal transduction pathways, there is a chain of reactions which transmits the signals of cell surface towards the intracellular targets. It was first studied for epinephrine, and it signals the glycogen breakdown to glucose. So this is the man who invented this pathway, who studied this pathway. And the concept is that CAMP is a second messenger. Please remember this line, CAMP is a second messenger. So CAMP signal transduction pathways. So the epinephrine receptor is coupled to adenine cyclic via a G protein, right? And then there's increasing the concentration of CAMP. Now, number one is the metabolic regulation. So let's talk about the process overall. So metabolic regulation. So the cytosolic protein kinase activation causes tetramer of regulatory and catalytic subunits, that is R2C2, which is inactive. The CMP binding of the receptor causes the dissociation of catalytic subunits and makes it active. Now, a serine threonine kinase causes activation or inactivation of substrate proteins. Now, the phosphorylation of two downstream enzymes are there in CAMP signal transduction pathway. The glycogen synthesis is inactivated, which decreases the glycogen synthesis, and the phosphorylase kinase is activated, which increases or which phosphorylates the glycogen phosphorylase, which increases the glucose 1 phosphate. So secondary messenger system. So the PLC, which is phospholipase C, gamma binds receptor protein tyrosine kinases via SH2 domain. And then it makes the domain phosphorylated, okay, or it makes the domain active. So it stimulates the hydrolysis of PIP2, which is phosphatidyl inositol biphosphate, to inositol triphosphate. So the diacylglycerol and inositol triphosphate are the secondary messengers here again. And the inositol triphosphate then regulates the calcium, and the diacylglycerol activates the PKC family. The diacylglycerol then remains associated with the plasma membrane. You can see remains associated with the plasma membrane and activates the protein serine or threonine kinases of the protein kinase C family. Remember the protein kinase that we talked about. And then the inositol triphosphate, which is a small polar molecule, is released into the cytosol. And now we know that the inositol triphosphate regulates the calcium. So it stimulates the release of calcium from the endoplasmic reticulum by binding to receptors that are ligand-gated calcium channels. The calmodulin is activated when the calcium concentration increases. And then the kinase family are activated by calcium calmodulin complex, and they phosphorylate and activate other proteins such as protein kinases, phosphatase, metabolic enzymes, and then the other metabolic processes go on to take place. This is how secondary messenger system works. So the non-muscle cells and the smooth muscles contraction is regulated by the phosphorylation of myosin light chain, and it is catalyzed by the myosin light chain kinase, which is regulated by the calcium binding protein calmodulin. Now you understand that how all these receptors, all these signaling molecules, all these pathways are actually involved in the contraction and other metabolic processes inside the body. So the increased calcium signals then causes further release of calcium from the intravascular reticulum by opening the calcium channels. And these calcium channels are called as ryanodine receptors in the endoplasmic reticulum membrane. Please remember the name of these receptors. Now, calcium is a versatile second messenger that controls a wide range of cellular processes. Now, these pathways function coordinately to regulate many cellular responses. Now, this one is also a very important pathway, which is called as the JAK-STAT pathway. Now, it causes the direct signaling from receptor to the nucleus. The ligand is cytokine, and the receptor is Janus kinase or non-receptor protein tyrosine kinase. STAT is signal transducer and activators of transcription. So the transcription factors contains SH2 domains that mediate the binding to phosphotyrosine sequences. The starts can be activated, they can be dimerized, or they can be translocated to the nucleus. And then these activate the transcription process. Thank you for watching. Keep studying hard.